and good afternoon in UK, and good morning in USA, and really early good morning in Australia, I suppose, if there's somebody coming from Australia. So this is so exciting. My name is Atharinam, and I take care of PASBAN, and we are just so excited to start this webinar series. um, um, by the first speaker is Professor Zafar Bashir. So let me tell you guys about PASBAN. PASBAN is the Pakistan Society of Basic and Applied Neuroscience. And the whole purpose of this society is to promote neuroscience in Pakistan. We want all those who are interested in neuroscience to come together, whether they are aspiring neuroscientists or they are neuroscientists who have already started to perform good research and establish themselves in institutions. So this is PASBAN provides that platform. PASBAN does on a regular basis an annual neuroscience conference, and we have been doing it now for the last 7 years. And this year, inshallah, it will be 8 ANC, 8th Annual Neuroscience Conference. We also have been taking care of our dear students by giving a series of lectures called Essentials of Neuroscience course. And ENC 2022 will be sometime in July and August, and that announcement is going to come. So please don't forget to check the website of PASBAN Neuroscience. This was formed in 2008. It stayed dormant for a while, but now we are very active and we have a lot of members coming in, and the membership have started. And for that purpose, our website, which is still in the forming phase, is going to give more information. So the most exciting thing that has happened in 2022 is that we have started the Neuroscience Educational Webinar Series. What we realized that there's a lot of talks on the YouTube, and a lot of talks, webinars are going on about neuroscience. But then what's really missing is for us students in Pakistan to understand neuroscience from the basics, how do you do research in that, and have a good interactive discussion. So that was the purpose of starting this webinar series, which is called Neuroscience Educational Webinar Series. The interactive part is the best part about it. And we are lucky to have the director of that news, Professor Zafar Bashir, who will be kicking off this news webinar series by actually giving his lecture first. Now, Zafar Bashir himself is a very, his story is very inspiring. He was born near Faisalabad in Chak number 204. So, that's, if those people who live in Punjab, they understand what it means really, right? And from there, he lived in Sindh near Dadu. And then he moved to London. And when he was 6 years old, he did his bachelor's in University of Newcastle, BSc in physiology, and then he did his PhD in Glasgow, University of Glasgow, and his work was on epilepsy model in rats. He is a very successful neuroscientist. I wish I could do things that he has been doing, and currently he's studying memory using some innovative techniques, optical conic model, which he will talk about, as well as looking at the NMDA receptors and other interesting things. So this is something that I would like you to see. If I can, right? Okay, here we go. So this is the logo of PASBAN, Pakistan Society of Basic and Applied Neuroscience. And there's a reason that why it is purple, because purple is a beautiful color. First of all, it's a royal color. And the purple is because, where I got my neuroscience training, that was purple. So, if Zafar had started this, then he would have made it red, because he has the University of Bristol, which is red color. The Twitter handle is right over here, PASBAN_group. And whenever you talk about in the social media about this webinar series, please remember to use this hashtag news 2022. That will be wonderful, because we want to spread the word around so that more and more people come and enjoy this thing. And I can't finish talking without mentioning the group that is so favorite to me, and that's NSIG. and that's a neuroscience interest group. This is a group that was formed by the students in AKU. Fatma Gauhar is right here with her screen on. She is the current president, and this group has reached out to a lot of other institutions across the country and beyond the country to develop a network of ambassadors. One of those brilliant minds in this group is Iza Tahir. Iza is going to moderate this session, and she's a third-year student at AKU, and she comes from Gujranwala, far out distance from AKU, and she is at AKU trying to get the best out of the best institution in Pakistan in medicine. So she will be moderating that. I think I have said enough, and I will hand it over to you, Iza. Thank you.
Thank you, Dr. Azhar. So, Assalamu alaikum, everyone. The webinar today will begin with a 45-minute presentation by Dr. Zafar Bashir, followed by a 15-minute question and answer session and a 20-minute general discussion. If you have a question, you are encouraged and requested to raise your hand on Zoom and unmute yourself when I call your name out. You are requested to also turn on your video to make this webinar more interactive. You can otherwise send in your questions in the chat box as well. You can also circulate the link of this talk to the session among your colleagues who might be interested in joining. I'd like to emphasize that for certificates, it is essential to fill out the feedback form, and the link of the feedback form will be provided to you at the end of this session. Dr. Zafar Bashir, you can begin now. You have the floor.
Thank you very much indeed. So first of all, just to say thank you to everybody who's been involved with setting up this seminar series. I know it's been a lot of work for a lot of people, and right at the very end, I was asked if I would like to be part of it and be the chairman of the news series, and I was happy to agree to that because actually all the work had been done for me. So to everybody who was involved in setting up this series, that's a fantastic achievement, and let's just hope that it goes from strength to strength. As was said right at the beginning, this is a means of transmitting and encouraging the generation of ideas in neuroscience in Pakistan, with people both in Pakistan but from outside of Pakistan as well, and hopefully encourage generation of ideas, discussion of neuroscience, and encourage people to think more about neuroscience as a potential future careers for those of you who are just starting out on your careers. And for those of you a little bit more long in the tooth, perhaps think about collaborations as well, how collaborations may be established with people both inside Pakistan but outside of Pakistan as well. So hopefully this will stimulate ideas, stimulate discussion, and there are a whole series of speakers that will be set up in the coming months from different parts of the world, but also from Pakistan as well, from amongst yourselves as well. So I would encourage you, as already been said, to spread the word about the seminars because it will provide information about the type of work that's going on out there, both in Pakistan and outside of Pakistan as well, the type of research that's being done, the kind of techniques that can be used, and hopefully you'll get a really broad sense of the type of neuroscience research that's happening, and encourage you to think more about neuroscience in your future careers as well. So the other thing I just wanted to say was I don't really know the kind of breadth of people who are going to be in the audience. I assume there's going to be a whole breadth from students, undergraduates, medical students, to those who are well established in the field as well. So I'm aiming to try and start off the presentation with a little bit of an introduction to synaptic transmission. I hope I won't be talking down to anybody, and I apologize if I do, but it'd be useful just to start, I think, with a little bit of an introduction. So that's kind of all I wanted to say right at the beginning, other than I just remembered that I forgot to say salam to everybody, so my apologies. Assalamu alaikum, everybody. And it's a real pleasure and an honor also to start off this seminar series by being the first speaker. So things can only get better from here. So let's see how it goes. Right, so share my screen. The right one, this one, I think. This one. Okay. So hopefully everybody can see that.
Yes.
Excellent. Pointer, let's get it. Right, brilliant. So what are we going to do now? What we're going to talk about is the research that I've been involved in for a number of years now, which started off, and I'm not going to talk very much about that, but started off looking at neuronal plasticity, synaptic plasticity. So that's trying to understand synaptic transmission. So that's how do neurons talk to one another within the central nervous system, and how is that communication modifiable? So that essentially is synaptic plasticity, neuronal plasticity. And then more recently, I've been interested in trying to understand how synaptic and neuronal plasticity can tell us about understanding mechanisms of learning and memory. So that's essentially what I'll try and talk through this seminar and try to give you an idea about the kind of work that we're doing in my lab. So before I start talking about the actual research, I just want to do a little bit of an introduction. So this image here, as you can all recognize, is an image of the human brain. The front over here, the back over here. Two areas that I'm going to concentrate on through my talk are this region here, which is the temporal lobe, which contains a really important part of the brain when it comes to learning and memory, which is the hippocampus. And I'm also going to talk about this region here, the medial prefrontal cortex, which is in the frontal lobes. Now, we know from lots of work that these two regions are very important in learning and memory. Now, what we don't know so much about is really the underlying mechanisms of how these regions are important. And that's the kind of work that I've been interested in, trying to uncover, and that's the kind of work that I'm going to be talking to you a little bit about today. So first of all, how do we know that regions such as the hippocampus and the prefrontal cortex are actually involved in learning and memory? And in many ways, actually, this is only a very recent, relatively recent finding. Knowing that there are specific regions such as the hippocampus are involved in learning is something that's only become about in the last few decades. So only sort of the turn of the middle of the last century do we actually begin to appreciate that specific regions of the brain, such as the hippocampus, actually have specific roles in learning and memory. So this is a classic example. So this is the work that resulted from surgery that was conducted on a patient called HM. This patient died about 15 years ago, and after that we knew his name, which is Henry Molaison. So in the States, this chap, Henry Molaison, had severe intractable epilepsy. And in the end, it was decided by his neurologists and his surgeons, this was back in the 1950s, so nearly 70 years ago now, that what they would do, the only way of relieving this epilepsy was actually to conduct surgery, which was to remove the hippocampus and parts of the temporal lobe. So this is one MRI scan from HM showing the regions where the surgery was carried out. Now, if you take a number of these MRI scans and put them together, what you can do is you can look to see how much of the temporal lobe and hippocampus was actually removed. And this is what's shown here on the left-hand side. So this is a normal brain. And this is HM's brain. You can see here how much of the hippocampus and temporal lobes were removed. All of the hippocampus on both sides, and significant parts of the temporal lobe, including parts of the perirhinal cortex, and parts of the amygdala were actually removed. And you can see that in this section here and this section here. Now, in terms of the epilepsy, this was a fantastic result because essentially the epilepsy was cured. So HM was essentially cured from his epilepsy. But the downside of the surgery is shown here. And this is was that HM was unable to make any new memories. And this is called anterograde amnesia. So memories that HM had prior to the surgery were actually intact. But anything that happened after the surgery, HM couldn't make new memories. So if he met a new person after the surgery, obviously that would be a new person. If he met that same person again an hour later, he would have no recollection that he had met that person an hour earlier. Now you can imagine that actually that's a very problematic for living a normal everyday life. Not being able to remember anything new and make new memories is actually very difficult. Makes your life actually very, very difficult. But what it told us for the first time actually that there was regions within the brain that were specific to specific types of memory. And this is what brought the hippocampus really to the forefront in terms of understanding memory, but also understanding how synapses work and synaptic transmission works. So those kind of experiments, if we can call it an experiment, that kind of experiment tells us a little bit about, if you like, a macroscopic understanding of a particular brain region is involved in a particular type of learning. It doesn't tell us anything, though, about how the memory or the learning actually occurs. So what we're interested in is a cellular understanding of memory. What's actually going on at a cellular level? What's happening at a synaptic level that actually allows memory to occur? This image here, this is an image which is taken from mouse brain, and this is about 2 millimeters by about 2 or 3 millimeters in each direction. This doesn't show all of the cells within this part of cortex, it just shows some of the cells, and these are color-coded according to their different functions. But what this shows is the complexity of a very small bit of brain, in this case here, just about 2 millimeters squared of mouse cortex. Now, when it comes to understanding how this bit of cortex works, what you've got to understand is how the synapses work. So how are neurons actually communicating with one another? Because what we believe is that actually it's the communication between cells that's actually important for understanding how memories work and how learning works. Now, it says down here, it's an interesting fact that each of these neurons may be connected to 1,000 other neurons. And if you do the maths, that means that in a brain, in a human brain, there could be something like 1,000 trillion synapses within the brain. Now, it's going to be impossible to understand what's going on at each of those 1,000 trillion synapses in the brain at any one time. So what we have to try and do is try and simplify things down a little bit to understand how synapses work and what that can actually mean for things like learning and memory. So if we want to understand synapses, here's a very simple schematic of a synapse. So this is a neuron, a pyramidal neuron within, let's say, the brain, within the cortex. And this pyramidal neuron provides an axon here. This is the axon from this pyramidal neuron, which is then forming an axon terminal, a bouton here. And that's forming a synapse with this next neuron. And this is the postsynaptic neuron. So this neuron here is forming a synapse via its axon bouton onto this postsynaptic cell here. And when an action potential travels down this axon, this causes a release of transmitter from these vesicles, and that transmitter is released into the cleft here, and that then acts on receptors on the postsynaptic side, so these postsynaptic receptors. And that essentially is the basis of synaptic transmission. So in some senses, it's a very, very simple process, and that forms the communication between one cell and another. And this is going on constantly in thousands and thousands and millions and billions of synapses in our brain at any one time. So this is what's occurring when you're listening to me. Hopefully, this is occurring in your brain. It's laying down some memory of what I'm talking about, so you can remember some of this later on. So let's just go back to the hippocampus now. So this is a cross-section through the hippocampus. This is actually a rat hippocampus, but the structure of human hippocampus is essentially exactly the same as the rat hippocampus. Now, one of the reasons people love working on the hippocampus is because of its known involvement in learning and memory, but also because of its beautiful structure, this very regular structure here. And essentially what we've got to have here, these dark patches here, these dark lines, are very dense collections of different cell types here and then through this region here. So this forms a very nice structure to actually work on. Now, if we lay on top of this just a cartoon which illustrates what's going on here, what we have in this region are a collection of cells, these are granule cells, and these form the dentate gyrus within the hippocampus, this region which is here. Now, the cells of the dentate gyrus, these granule cells, they send their axons, as you can see here in red, via what are called the mossy fibers to the CA3 region of the hippocampus. And these make synapses onto the dendrites of the CA3 pyramidal cells. Now, the CA3 pyramidal cells here, they have axons which then project via this region here, which is called stratum radiatum, and they make synapses onto the CA1 pyramidal cell dendrites. So what we have is a beautiful little system here where you have input coming from the entorhinal cortex, makes synapses onto the granule cells in the dentate gyrus. These make synapses onto the CA3 pyramidal neurons. These in turn make synapses onto the CA1 pyramidal neurons. So this is a classic trisynaptic circuit within the hippocampus with an input and an output which goes from the CA1 hippocampus to a bunch of different regions. Now, the hippocampus has been used extensively over the last decades to try and understand how each of these synapses work and what the function of each of these synapses is in different normal behavioral functions. So if you want to understand synaptic transmission, this is a very simple system for doing this. What you can do is you can place a stimulating electrode down here in the axons of the CA3 axons down here. If you stimulate these axons, these will fire action potentials, and this will cause synaptic transmission that you can then record in the CA1 pyramidal cells. Now, if you do that, so this is a recording here, this is showing a recording electrode in the CA1 pyramidal cell. So you can do recordings from individual CA1 pyramidal cells, as is shown here. And when you stimulate these axons, what you can get is a synaptic response, which is shown here. So this would normally in the cell be a depolarizing potential. This would be an excitatory postsynaptic potential. In this case here, this is a voltage clamp recording. So what you're recording here is the underlying current that would be produced when these axons are activated, and these produce EPSPs in the dendrites where these synapses are located. So this is the synaptic response that occurs when you stimulate the axons onto this cell here. So what you can use this for is to understand what the mechanisms of synaptic transmission are. You can work out the transmitters, receptors that are involved. You can work out what transmitters may modulate this response. You can work out the plasticity mechanisms of this response as well. So you can do a whole range of interesting experiments to understand the synaptic physiology of transmission at each of these different synapses. Okay, so let's have another look at synaptic transmission. Here again is our presynaptic neuron with the presynaptic bouton here. It's releasing transmitter into the synaptic left onto the postsynaptic neuron here. This is shown in a little bit more detail. Here we have the synaptic vesicle fusing. It's releasing in green the transmitter, and the transmitter is acting on these postsynaptic receptors here and here. When the transmitter acts on these receptors, it opens the channel in the receptor. This particular receptor is permeable to this ion here, which might be sodium in red, and the sodium will flow into the postsynaptic cell, into postsynaptic dendrite, and depolarize the postsynaptic cell. And that's what produces this synaptic response here. Now, in most central nervous system synapses, the transmitter here in green is glutamate, a very simple amino acid. So in most excitatory synapses in your brain, the transmitter being released here is glutamate. And glutamate will bind to these postsynaptic receptors. And that's a very simple way of producing synaptic transmission. Now, the complexity of synaptic transmission occurs because glutamate doesn't just bind one receptor type, it binds lots of different receptor types. And these are a combination of ionotropic receptors, grouped here, NMDA receptors, AMPA receptors, kainate receptors, and delta receptors. And these are linked directly to ion channels. So you'll get an ion flow when you activate these receptors here. And they're also metabotropic receptors. And these are G protein coupled receptors, which link to intracellular cascades. And so they will activate intracellular calcium levels, and that will cause activation of, for example, protein kinases and phosphatases. And so even though you only have one transmitter, glutamate will act on lots of different glutamate receptors, and that will have lots of different functions in a given synapse. And the beauty of this system here is that different synapses in different parts of the brain will have different ratios, different complements of these receptors. So when glutamate is released, depending on where the synapse is, in which particular part of the brain, the actual response that's produced here will depend on the complement of these different receptors, and it will be different in different parts of the brain. The other complexity I should also add is that if just take, for example, the NMDA receptor is composed of different subunits, and you'd have different complements of different subunits in different synapses. So the synapse, even though it looks very simple when you think about it like this, and you have one transmitter, actually the response that's produced can be very, very different under different conditions, in different cell types, in different parts of the brain. And also on top of that, you have different ratios of these and different complements of these receptors during development. So early in development, you'll have different combinations of these, for example, NR1, NR2, 2B receptors. Later on in age, you have different ratios of these subunits of receptors, and that will actually produce very different functions in the synapse. With different diseases also, you find that it changes also the combination and the complement of these receptors. And with synaptic plasticity, when synaptic plasticity occurs, that will also change the complement of these receptors as well. So often we think about synaptic transmission as just being a very simple process where all we have is an EPSP, and that's the outcome of transmission. But actually this EPSP, the regulation of this EPSP, the modulation of this EPSP, the way this EPSP changes with development, with plasticity, with disease, a lot is going on to control and regulate this EPSP through here, just different types of glutamate receptors. On top of that, which I'll talk about later on, we have other transmitters which will also act on these synapses, which will also act to regulate glutamate transmission as well. So the synapse, in one way, we can think of it as just being a very simple gate, which will produce a response, but actually that gate is subject to very complex interaction and very complex regulation. We'll talk a little bit about that later on. Okay. Right, so that's an introduction to synaptic transmission and a little bit about the basics of how we know a little bit about learning and memory. What I want to now talk about and focus on is something about some of the work that we've been doing in my lab, thinking about the neuronal and synaptic basis of learning and memory. The work that I'm going to focus on now is some work that we've done in the last few years, which is looking at acetylcholine receptors, specifically nicotinic receptor subtypes. How those nicotinic receptors regulate excitatory transmission between the hippocampus and prefrontal cortex, and how that brings about synaptic plasticity, and then how we think that modulates and brings about different phases of associative recognition memory. So there's quite a lot to take in there, and I'll try and go through this quite slowly in the next set of slides. This work has been done in conjunction with Marie Sebeck, who is a PhD student, who is now a postdoc in the lab, and in collaboration with Sue Waddicott, who's at Bath University, and in collaboration with Claire Warburton, who's a PI here at Bristol University. Okay, so what do we mean by, or what do I mean about associative recognition memory? So associative recognition memory is remembering a scene such as this, for example. So if we look at a scene like this is actually a bike park in Amsterdam, not in Bristol. I cycle to work every day, and when I cycle to work, I park my bike in not quite such a busy bike park as this, but nevertheless, I park my bike in the bike park. Now the question is, when I come along back at the end of the day to pick up my bike, how do I know where my bike is? How do I know where I left my bike? Now, one thing is very simple. Let's just say this is my bike here. I know my bike. I've been riding it for many years. So that's a single item. You can treat your bike as a single item. And you remember single items through processes that occur in the perirhinal cortex, which is part of the temporal lobe. And so we've been doing work for some years on the perirhinal cortex and how the perirhinal cortex allows us to understand single item recognition memory. It's an automatic type of recognition memory. I'm not going to talk about that now, so just take my word for it. The perirhinal cortex is involved in me knowing that this is my bike. Now, I park my bike, I come along at the end of the day. How do I find my bike? The way that we do that is we use associative recognition memory. So there are various landmarks around here in this scene here. And so what the brain does, again, this is a very automatic process. I don't have to think about, I must remember that my bike is somewhere near this lamppost. I must remember my bike is near this tree over here. I must remember that my bike is next to this car over here. We don't stand there having to remember these things. Again, this is a very sort of automatic type of memory, but it's an associative recognition memory. I remember where my bike is by using these various landmarks, by associating my bike position with these various different landmarks over here. And this is a very normal automatic type of memory, and we use it every single day in our normal everyday lives, and we do whatever we do in our normal day. Now, we know different regions of the brain that are involved in associative recognition memory, and there's lots of work that's gone on in this sphere, not just work that we've done, but lots of work that's going on around internationally, looking at associative recognition memory. We know the medial prefrontal cortex is key, the hippocampus is key, parts of the thalamus, such as the nucleus reunions, and the medial dorsal nucleus of the thalamus and other regions are absolutely essential for associative recognition memory. So if you knock out the medial prefrontal cortex, you have deficits in associative recognition memory. If you knock out the medial dorsal thalamus, you get deficits in associative recognition memory. We've also been recently looking at the connectivity between these regions as well. So we know now the hippocampus connections to the medial prefrontal cortex are essential. We know connections from, for example, the lateral entorhinal cortex to the medial prefrontal cortex are essential for associative recognition memory. So we've been doing quite a lot of work on looking at these connections between different regions and which ones are important and how they're important. What I'm going to talk about today is just work that we've been doing looking at the connections between the hippocampus and the medial prefrontal cortex, and how those are important, and how plasticity between these connections might be important for associative recognition memory. Okay, so the work that I'm going to describe is all carried out in either rats or mice. Now, the way that we assess associative recognition memory in rodents is shown in this task here. So basically what you have is you have a sample phase and a trial phase, in which the encoding of this, this collection of objects is carried out, and then the retrieval of the memory is carried out sometime later, in this instance 3 hours later, but it can be any time span. So what you do is you take a rat, in this case here, you put it in an arena with 4 different objects in this particular arrangement. The rat is allowed 5 minutes to explore these objects. You then take the animal out, give it 3 hours, and you put the animal back into this arena. But now you may notice that 2 of these objects have switched place. Now, what the normal animal will do is spend more time exploring those objects, those 2 objects that have switched place, compared to the 2 objects that have stayed in the same place. And it will do that because it remembers the original association, it remembers the association of these original location of objects. So it remembers those, and it will notice that these 2 have changed place, and it will spend more time exploring these 2. So essentially what the animal is interested in is the novelty that occurs in this phase here compared to here. And we as humans do that as well. When we encounter something like this, we spend more time exploring and looking at the novel arrangement of items compared to the familiar ones. What we simply do then is just tot up the time, add up the time the animal spends exploring these novel arrangement of objects versus the familiar arrangement of objects. We take a ratio of those, and we express this on a histogram like this, a graph like this, which we've called here the memory strength. Anything that's significantly above 0 means that the animal has now remembered this original sample phase this is original phase, sample phase over here. And so that's a measure of memory strength. And so what we look for, if there's a deficit in memory, is a decrease in this discrimination ratio, or memory strength, as it's called here. So that's how we do most of the behavioral experiments that I'll be describing. So that's what's being described here again. This is the same kind of experiment. Ignore for now this laser on, laser off. I'll come to that in a minute. But essentially what we do is the same experiment here, sample phase and test phase. We run the controlled animals through this, and the animal performs the task perfectly well. And it's remembered the original, arrangement of these items here when you put it in the test phase over here. It spends more time exploring the moved objects compared to the unmoved objects, and that's what is shown here. So in these experiments, what we're doing is we're looking at how important the connections between the hippocampus and the prefrontal cortex are for this type of associative recognition memory. Now, what we've done in the experiments here, we've disconnected the hippocampus to the prefrontal cortex. And the way that we've done that is to use these optogenetic methods. And so what we do is we use this viral technique to express this inhibitory opsin arch in the hippocampus. And so use this adenoviral construct, which is expressing arch in CA1 pyramidal neurons in the hippocampus. So you inject this construct into the hippocampus. This construct is taken up by hippocampal pyramidal neurons. It gets expressed in the genome of those pyramidal cells, and those pyramidal cells then express arch, and arch is also linked to this yellow fluorescent protein here. And so the hippocampal pyramidal cells take up this protein. It's then expressed in the pyramidal cell bodies, but it's also expressed throughout the axons of those pyramidal cells, wherever those pyramidal cell axons project to. This image here is from the prefrontal cortex here. So this is the prelimbic cortex and infralimbic cortex, and what it shows is it shows expression of the EYFP, the yellow fluorescent protein, which is showing as green here, but that's not important. So this is a fluorescent protein which we're using as a marker to show that we're getting expression of this construct in the pyramidal cells and right throughout importantly, right throughout the axons which project to the medial prefrontal cortex. So you can see the projection of this in the prefrontal cortex. What we then do is put an optrode into the prefrontal cortex, which is shown here and here, and we can pulse laser pulses via these optrodes into the prefrontal cortex. And the reason that we do that is those laser pulses will then activate ARCH. And ARCH, this protein here, which we're expressing in these axons, is a proton pump. And when that's activated, what that does, it hyperpolarizes those axons and essentially will depress synaptic transmission only at these axons from the hippocampus in the prefrontal cortex. All the other hippocampal projections to other parts of the brain will carry on working as normal. All we're doing now is preventing that transmission here in the prefrontal cortex. So the hippocampal to the prefrontal cortex projections are silenced when we activate this laser pulses into the prefrontal cortex. So what we do is we do that experiment while the animal's actually doing the behavioral task. And we've done that here during the sample phase. So what we do is turn the laser on during the sample phase here. Then what we do is we give the animal a 3-hour delay, put the animal back into the test phase and test to see how much time the animal spends exploring these pair of objects and these pair of objects. And as you can see here, when you turn the laser on during the sample phase and then do the test phase, the animal now spends equal amount of time exploring this pair of objects and this pair of objects. So what that's telling us is when we turn the laser on and inhibit transmission between the hippocampus and the prefrontal cortex during the sample phase, this animal hasn't made this memory for this arrangement of these different objects during the sample phase, because it doesn't now recognize that the original arrangement of items as the control animals do that don't get laser stimulation. So in the same animals that have no laser stimulation during the sample phase, the animals perform the task perfectly normal. They spend more time in the test phase exploring these pair of objects compared to this pair of objects. So what we've done by silencing this projection here is we've prevented the encoding, we've prevented the learning of this arrangement of objects. So that shows that the hippocampal to prefrontal projection is essential for object in place associative recognition memory. This is just to show that this is a separate experiment showing that you can that we're actually silencing action potential firing in these pyramidal cells. So here's a control recording where the cells will fire action potentials, but when you turn the laser on, there's no action potentials being fired. So that's just a control showing that what we expect to be happening when we activate the arch is to silence these projections. That's just a control here showing that we can silence those, that we are actually silencing those cells from firing. Okay, so now what we're interested in, having shown that those hippocampal to prefrontal projections are essential for associative recognition memory, what we're interested in is understanding how do nicotinic acetylcholine receptors regulate transmission between the hippocampus and prefrontal cortex? And what's that got to do, if anything, with associative recognition memory? So this is just a schematic showing the projections of the cholinergic projections from these regions here, the PPT and the LDT. So that's the pedunculopontine tegmentum, which projects acetylcholine to various different parts of the hindbrain, the brainstem, the cerebellum. But the regions that we're interested in, this nucleus basalis projection, this is another projection, cholinergic projection, and that projects extensively to the cerebral cortex, which is what we're interested in here. So these cholinergic projections project to the cerebral cortex and the frontal cortex, the prefrontal cortex. So we know there are heavier projections from the nucleus basalis to prefrontal cortex. This slide here just illustrates the two nicotinic acetylcholine receptor subtypes that we're particularly interested in this project. Those are the alpha-7 nicotinic receptors and the alpha-4 beta-2 nicotinic receptors. And those are heavily expressed within the cerebral cortex. So there are lots of alpha-4 beta-2 receptors, alpha-7 nicotinic receptors, and also these other alpha-4, alpha-5 beta-2 receptors as well. We're going to focus on the alpha-4 beta-2 and the alpha-7 nicotinic receptors for the purposes of this. So in these particular experiments, what we're going to do is we're going to run the same type of behavioral experiments that I've described already. But what we can do is we can now inject into the prefrontal cortex antagonists of alpha-7 or alpha-4 beta-2 receptors at different times during the behavioral tasks. We can inject those inhibitors prior to the sample phase, and that will disrupt the acquisition or the encoding phase. And we can inject those inhibitors immediately after the sample phase to interrupt the consolidation of memory, or immediately prior the test phase to disrupt the retrieval of object-in-place associative recognition memory. This image over here just simply shows the cannulae, so this is post-experiment, after we've done the experiments, and we just check to see where the cannulae were. So the injections are done by having indwelling cannulae within the medial prefrontal cortex, and those can be embedded within the prefrontal cortex, and you can run these experiments over several months and inject various different inhibitors into the prefrontal cortex at various times over the course of several months during these experiments. So the first experiment here is testing the role of alpha-7 nicotinic receptors in associative recognition memory using this antagonist called MLA. And so these experiments here are the same type of experiments I've described before. We're measuring discrimination ratio, which is a measure of memory strength. And what we've done is injected the antagonist MLA to block alpha-7 nicotinic receptors at various different times, like I described in the previous slide, either testing the effect of these receptors on acquisition, consolidation, or retrieval of associative recognition memory. You see from this slide here, when we inject the antagonist to disrupt acquisition, you can see that that completely blocks the acquisition of associative recognition memory. It has no effect on the consolidation phase, and it has no effect on the retrieval of associative recognition memory. So activation of alpha-7 nicotinic receptors are essential for the acquisition or the encoding of associative memory, but are not required for consolidation or retrieval. We've done the same experiment with this compound with DHBE, which is an antagonist for alpha-4 beta-2 nicotinic receptors. So we're blocking the function of alpha-4 beta-2 nicotinic receptors again to block those receptors during acquisition, consolidation, or retrieval. And what this data shows you is when you block alpha-4 beta-2 receptors, this has no effect on acquisition, no effect on consolidation, but will prevent the retrieval of associative recognition memory. So the activation of alpha-4 beta-2 nicotinic receptors is important for long-term associative recognition memory retrieval, but not for acquisition or consolidation of associative recognition memory. Okay, so that tells us something about the receptors that are required within the medial prefrontal cortex for different phases of memory. It doesn't really tell us very much about the synaptic processes or plasticity processes that are occurring in the medial prefrontal cortex. So that's what we're interested in the next few slides. So what I've shown you also is that the hippocampal to medial prefrontal cortex projection is important for associative memory. I've also just shown you that alpha-7 nicotinic receptors and alpha-4 beta-2 nicotinic receptors are important for different phases of associative recognition memory. So we wanted to understand something about the synaptic mechanisms between hippocampus and medial prefrontal cortex and how those might be modified by activation of nicotinic receptors and how that might contribute to learning and memory. So the way we do these experiments is we take slices of medial prefrontal cortex, much like the experiments that I described with the hippocampal slices. So we take slices of medial prefrontal cortex as is shown here. We take recordings from individual pyramidal neurons as shown here within medial prefrontal cortex, and we can stimulate the hippocampal projections that come into the medial prefrontal cortex. And then we can record synaptic responses, as is shown here, within these medial prefrontal cortex pyramidal neurons in response to the hippocampal input that comes in and makes synapses onto these cells. Now, we're interested in understanding the plastic mechanisms that occurs at this hippocampal to medial prefrontal cortex synapse. Now, in order to induce plasticity of these synapses, this is a very classic protocol that's used. So what you do is you pair the postsynaptic firing of action potentials in the pyramidal cell with presynaptic firing in the hippocampal projection. And that produces, in many different synapses in the brain, that produces a form of plasticity called long-term potentiation. And long-term potentiation has been thought of for quite a few years now as being a really important mechanism in bringing about the mechanisms that might underlie learning and memory. So this is just a protocol that's induced. It's a protocol that's been shown in many different synapses to produce LTP, and LTP is thought to be important for some of the mechanisms of learning and memory. Now, if we deliver this protocol to the synapses that we're studying here, which is the hippocampal to medial prefrontal cortex synapses, shown here, the arrow here indicates where we deliver this protocol. So this is a protocol, we pair 5 EPSPs, shown here, with 3 action potentials in the postsynaptic cell, and it's repeated 80 times at theta frequency. And it's repeated at theta frequency because theta frequency is a very robust frequency that occurs in the brain when we know that learning and memory occurs in the brain. That's been shown in a whole range of experiments in different animals, including in humans as well. So theta frequency, repetitive activation, is very important for learning and memory. Now, if we do that experiment, what we're doing here is, as I've just said, is we're recording EPSPs in the postsynaptic pyramidal cell, giving this protocol here, which is shown by this simple arrow, and we look to see what happens at the synapse. And essentially what happens at the synapse is we get nothing. All we get is a very small increase in synaptic transmission shown here. So on the y-axis, we have the amplitude of this response. We get a very rapid increase, but actually we get no long-term potentiation at all. So this synapse doesn't show long-term potentiation as we were expecting under these conditions. So this is the experiment I've just shown you. When we give that pairing protocol, it produces no plasticity, no long-term plasticity at this hippocampal prefrontal cortex synapse. So because alpha-7 nicotinic receptors are important in learning and memory in the prefrontal cortex, what we thought we would do then is to actually stimulate these alpha-7 nicotinic receptors at the same time as giving this pairing protocol and see what happens. What we found was that if you stimulate alpha-7 nicotinic receptors in conjunction with the pairing protocol, this is shown in orange, and the agonists, what we're doing is applying the agonist, this agonist compound here, which is called PNU, which is an alpha-7 nicotinic agonist, and that's the data shown here in orange. What we find is that produces now very robust long-term potentiation. And that long-term potentiation is blocked by the antagonist of alpha-7 nicotinic receptors, shown here in red. So if we activate alpha-7 nicotinic receptors, and that produces long-term potentiation of the hippocampal prefrontal cortex synapse. If we do the same experiment, but now with the alpha-4 activation of the alpha-4 beta-2 nicotinic receptor, and that compound is called RJR. We've used the alpha-4 beta-2 agonist, RJR. And that's shown in this sort of light green, I guess. What we get there now is long-term depression. So we've got a depression of synaptic transmission through activation of the alpha-4 beta-2 nicotinic receptor. And that long-term depression is blocked by the alpha-4 beta-2 antagonist, this compound DHBE, which is shown in the dark green. So activation of alpha-7 nicotinic receptors produces LTP at the hippocampal prefrontal synapse. Activation of alpha-4 beta-2 receptors produces LTP at the hippocampal prefrontal synapse. And you remember from the previous slides, activation of alpha-7 nicotinic receptors is crucial for the encoding phase of associative recognition memory, not for the retrieval phase. Activation of alpha-4 beta-2 receptors is required for the retrieval, but not for the acquisition of associative recognition memory. So that leads us to the question, is this bidirectional plasticity brought about through alpha-7 nicotinic receptors and alpha-4 beta-2 receptors required for encoding and for retrieval of associative recognition memory? I'm looking at the time, I'm realizing that time is running out very quickly, so I'll try and do the next part quite quickly, but also try and hopefully make it as clear as I possibly can. Right, so what we've been looking at in the previous slides is the induction of LTP and LTD through the activation of alpha-7 nicotinic receptors and alpha-4 beta-2 nicotinic receptors. So what we wanted to do was find an alternative way of blocking LTP and LTD without interfering with nicotinic receptors. This schematic here shows the processes, some of the processes that are involved in the expression of long-term potentiation. So here's our glutamate synapse. Glutamate acts on AMPA receptors to produce the normal EPSP. Now, LTP occurs when a whole set of processes occur in the postsynaptic cell. And those processes, what they do is increase the number of AMPA receptors in the postsynaptic synapse. And that increased number of AMPA receptors is what produces long-term potentiation, because you have more AMPA receptors, there are more AMPA receptors for glutamate to bind to, and that increases synaptic transmission. And that's essentially the basis of LTP. Now, one of the mechanisms that is important for producing or for stabilizing these AMPA receptors at the synapse is this kinase here, which is called PKM-zeta. So if we can interfere with PKM-zeta, we can prevent LTP from occurring without interfering with nicotinic receptors. Now, the way to do that is to use a compound which is called zeta inhibitory peptide. And zeta inhibitory peptide essentially blocks the functions of PKM-zeta and therefore should block LTP. So in this experiment here, this is another experiment looking at synaptic plasticity at the hippocampal prefrontal synapse. And what we've done in this experiment here is used the compound ZIP, which will block the action of PKM-zeta, in conjunction with the alpha-7 nicotinic agonist, which we've shown in the previous slides to bring about LTP. So if you look at the purple dots, what that shows is that if we do that experiment that previously produced LTP, but now in the presence of the inhibitory peptide ZIP, there is no LTP. So when we block the action of PKM-zeta, we block LTP. In the orange is the control data. This is just scrambled ZIP. This is ZIP is just a peptide, and if you scramble the sequence of amino acids in this peptide, then ZIP doesn't have the normal functions, so it doesn't block the activation of PKM-zeta, and we get normal LTP in the presence of the agonist of alpha-7 nicotinic receptors. Now, we can do similar experiments to block LTP. So this is just a schematic showing how LTD is produced. Now, LTD is essentially the opposite of LTP. So here we have synapse, and we have receptors, AMPA receptors in the synapse. And the way that LTD occurs is to remove AMPA receptors from the synapse. So we have fewer AMPA receptors in the synapse, which means there are fewer receptors for glutamate to bind to, and so the EPSP here will become smaller. And that's essentially the mechanism of LTD. Now, the way that these AMPA receptors are internalized into the synapse here, removed from the synapse, is through, shown schematically in this schematic here. So here's an AMPA receptor with a long C-terminal tail, and the endocytosis, the internalization of these receptors, happens through a fairly complex process which involves this protein called AP2. And what AP2 does is binds to amino acids on the C-terminal domain, and that binding is essential to internalize these receptors. So if we can interfere with this interaction between the C-terminal of the AMPA receptors and this protein AP2, then we can prevent LTD from occurring. And we can do that through using this peptide called GluR2-3Y. And that prevents the interaction between the AMPA receptor and AP2 and should prevent the induction of long-term depression. So again, an electrophysiology experiment shown here, looking at the hippocampus prefrontal synapse. If we look at the blue dots over here, these experiments are done in the presence of this GluR2-3Y peptide, which will prevent the internalization of these receptors. And when we do the experiment in the presence of the alpha-4 beta-2 agonist, RJR, we find that we can block LTD with this peptide. And then shown in this greeny color here is the control experiment with another version of this peptide, which doesn't block the interaction. In this case here, we get LTD. So we can block LTD, we can block LTP using these mechanisms here. So now the question is, what happens when we block LTP and LTD in a fully functioning, behaving animal in the different phases of learning and memory? So we go back to the behavioral experiments here with an animal which is running these tasks, as I've shown you before, and we can inject these peptides into the prefrontal cortex to block the encoding phase, or separately to block the retrieval phase. And when we put the ZIP peptide, which blocks LTP, into the prefrontal cortex, we find that this completely blocks the acquisition, the encoding of object-in-place associative recognition memory. But this ZIP peptide, which blocks LTP, has no effect on the retrieval phase of associative memory. And when we use the GluR2-3 Y peptide, which blocks LTD, you see that it has no effect on the acquisition, on the encoding of associative recognition memory, but it completely blocks the retrieval phase of associative recognition memory. So if we go back to thinking about associative recognition memory, as I described before, and the connections between the hippocampus and the medial prefrontal cortex, this connection between medial hippocampus and medial prefrontal cortex is an excitatory projection. It's the glutamatergic synapse between the hippocampus and the prefrontal cortex. But this glutamatergic synapse is being modified, is being regulated by the release of acetylcholine into the medial prefrontal cortex. So what we think is going on during the encoding of associative recognition memory, when I leave my bike here in the morning, I'm setting up the encoding of associative recognition memory. So the synapses between the hippocampus and the medial prefrontal cortex, this synapse here, onto these pyramidal cells in prefrontal cortex, are being modified by the release of acetylcholine. And that's happening through the activation of alpha-7 nicotinic receptors, which is causing LTP at this hippocampal to prefrontal synapse. And that LTP is essential for the initial encoding phase. When I come back in the evening to pick up my bike, what's going on is that this synapse here, between the hippocampus and the prefrontal cortex, this synapse here, is a glutamatergic synapse, but it's being modified by the release of acetylcholine, which is now acting on alpha-4 beta-2 nicotinic receptors. And we think this is acting on interneurons. These alpha-4 beta-2 nicotinic receptors are on interneurons, and this interneuron activity is what's crucial to produce then LTD, these synapses. And this alpha-4 beta-2 nicotinic receptor LTD is what's important for the retrieval phase of associative recognition memory, allowing me to come back in the evening to find my bike and in order to retrieve the association of these different components of the scene here so I can find my bike. So we think these are some of the different mechanisms that are occurring during the encoding and the retrieval phase of associative recognition memory. It relies on these different glutamatergic receptors, it relies on these different nicotinic receptors, all the different phases of associative recognition memory. Okay, so it's 2 o'clock now. I was going to talk a little bit about another aspect of work that we are doing here, but I obviously don't have time to talk about that. So I'm going to leave that maybe for another time, but that was going to be talking about specifically about the CA2 region of the hippocampus and how that's involved in social recognition memory through the action of metabotropic glutamate receptors and the interaction with these proteins here, which are specifically found in the CA2 regions of the hippocampus, which are called RGS14 proteins. These are regulators of G protein signaling, and they regulate metabotropic glutamate function and can work to bring about social recognition memory, and that is active within the CA2 region of the hippocampus. But obviously I haven't got time to talk about that now, so I will stop talking and just say thank you very much indeed for your attention, and I'm happy to take any questions. I'm sorry that we're running a little bit over time. Thank you very much.
That was just wonderful.